Hello everyone, the technique that we'll be talking about today is immunohistochemistry. But before we dive in, let's talk about some background information. So there's a couple things you need to know. First off, antigens are proteins and carbohydrates that are embedded in the cell membrane, which is a protective barrier that is on the outside of cells. And those antigens are there to help with cellular identification and signaling. While antibodies are proteins that detect specific antigens, which aids in marking and is useful for the immune response. Both antigens and antibodies fit together in a bind called lock and key binding, which pretty much means that an antibody can only bind with a specific antigen. Now that we have some background information, let's talk about immunohistochemistry, which is a scientific technique that uses antibodies to detect antigens on a target cell or tissue and tags it so that it can be seen and analyzed. As you can tell from the picture, there's a tissue that they want to analyze. Then there's a primary antibody that is attached to the target antigen on that tissue. Then, attached to the primary antibody, there is a secondary antibody that is connected to an enzyme that uses its substrate to make a colored product so that it can be seen for imaging. Although in this example, there is an enzyme that is being used to make the colored product, that is not always the case, but we'll talk about that later. The basic process of the immunohistochemistry is as follows. Step 1 is to retrieve antigens, 2 is blocking, 3 is putting on the primary antibody, 4 is then the secondary antibody, and lastly is imaging. But let's take a closer look at each step. So the first step is antigen retrieval. This step is to ensure optimal binding of antibodies to the target antigen. They do this by taking the sample of tissue that they want to test and putting it through intense heat or by using enzymes to break apart binds that might be masking or covering up the target antigen. This allows the target antigen to be more exposed and allow for optimal binding with the primary antibody. The next step is blocking, which is the step that scientists do to prevent unwanted binding between the secondary antibody to non-target antigens and other binding sites. This process ensures that the secondary antibody will only bind with the primary antibody, which would lead to a more accurate findings. They do this by using serums, which are essentially made up of a match of the secondary antibody, which allows the serum to bind to the non-target binding sites that would want to bind with the secondary antibody if otherwise. The next step is when they add the primary antibody onto the sample. The primary antibody binds to the target antigen and allows for there to be a binding site for the secondary antibody. Next, there's a secondary antibody, which is either paired with an enzyme label or a fluorescent dye. A secondary antibody with an enzyme label is used to make an enzyme use its substrate to make a dye that can be seen, while a secondary antibody, which is paired with a fluorescent dye, already has what it needs to be seen. Both types of secondary antibodies ensures that the target can be seen, and the secondary antibody binds to the primary antibody. Lastly, there's imaging, which is the use of fluorescence and light microscopes and other devices that are used to take pictures and to see the target antigens for it to be analyzed, quantified, or even just to admire. Immunochemistry can be used in many things, but to name a few, it can be used in experiments to figure out the concentrations of proteins in cells. It can also be used to diagnose cancers by detecting cancer cells, and it can be used to predict treatment responses by that it can identify cells that can affect treatment plans. Thank you for listening and have a great day. Here are some of my sources.